Today we will start with the immune system. In immune system, you will be studying about different type of immune activity that is taking place, how the body is defending against the disease, how the inflammatory process starts, various objectives you have. Let us start with the learning outcome. So, first outcome is describe the major site of immune cells production and development. Then comprehend three main lines of defense that has evolved to prevent infections. Understand how non-specific defense system involving phagocytes, lysozymes and NK cells works. Outline the process involved in inflammatory and allergic response, and also now how the non-specific immunity works in the body. Then types of immune responses, then hemolytic diseases, structure and function features of antibodies in the human body. Understand the difference and give examples of autoimmune and immunodeficiency disease. These are all the learning outcomes you will know. And coming to the immunity, what is immunity? It's a resistance to disease. Okay, now you know many people suffer with a lot of viral infections. So if there is good resistance in the body, then we are able to defend the viral infections. Okay, so in that case you have two types of immunity inside the body: the innate immunity and adaptive immunity. Innate immunity is mostly it's non-specific defense system. Adaptive immunity is specific type of defense system in the body. Okay, so as far as innate defenses is concerned, the skin is acting as a barrier which doesn't allow any bacteria to go inside the body, and the mucous membranes in the mouth, in the GI tract, then in the all parts of GI tract everywhere. Okay, then next is the internal defense system, that is the defending cells that is present inside the body, like the WBC cells, okay, natural killer cells, inflammatory reaction, then antimicrobial proteins, fevers, all these are innate defensive system. Then as far as adaptive defensive system is concerned, you have two types of immune cells. One is B cells and the T cells. So B cells is coming under humoral immunity and T cell is coming under cellular immunity. Okay, then what is this first line of defense? So we will discuss about the innate defense system. Okay, as far as skin and mucous membrane is concerned, in the skin you have lots of layers of the skills: stratum, corneum, lucidum, then spinosum, granulosum, basal, different types of skin layers. In that cells, there are the keratinocyte cells that is distributed all over the skin cells. Okay, so this keratin resists to weak acids and bases, bacterial enzymes and toxins. Mucosa in the GI tract that provides similar mechanical barrier. It doesn't allow the bacteria to penetrate inside the body. So the skin is a good protector organ. Skin is said to be the largest sensory organ. It is also a good protecting mechanism. It is also acting as a good protective mechanism. Okay, so this is a summary. You can see that we discussed that the skin that is preventing all the bacteria to enter, and intact mucous membranes which helps the mucus is trapping the microorganism, nasal hairs filtering the dust, then cilia, that is the hair like structures on the respiratory mucosa, is preventing the mucus to go to the alveoli. Then gastric juice kills the bacteria. Then acids environment in the vaginal cavity. Then lacrimal secretions on the in the eyeball. I mean in the optic cavity, so that they wash out all the bacteria and the dust. Then the urine, which naturally excretes all the metabolic waste. Okay, so coming to the surface barriers, produces protective chemicals, that acids. So different types of acids are produced. For example, stomach is producing this hydrochloric acid, This acidic environment is also present in the vaginal cavity. Then acidity of the skin, that is also killing the bacteria. Then enzymes. So in the saliva there is an enzyme called lysozyme. Okay, this is present in the respiratory mucus and also lacrimal fluid, that kills the bacteria. Okay, then the mucins. If you look at the mucins, it traps. The main function is to trap the dust and the organisms. Okay, then defensin is secreted in the mucous membrane and the skin, that increase, when there is inflammatory reaction, this defensin is secreted. It's a type of broad spectrum antimicrobial peptide. Okay, then other chemicals, dermcidin, is also excreted in the sweat gland. Okay, then the second line of defense, that is innate defenses, we have the cells and the chemicals. One is the phagocytic cells, okay, which the neutrophils and the macrophages, they go and swallow the bacteria. I will show how it swallows and how it getting neutralized. Then natural killer cells, for example, cells keeps multiplying, getting old, so these cells are being replaced. Okay, so then likewise inflammatory response. So any type of foreign body goes inside the tissue, like it causes inflammation and tries to expel this foreign body. So this kind of inflammatory response is natural. For example, any processes that we keep inside the body, it's a foreign body. Okay, so we don't use normal metal. This process is, for example, mitral valve or stents are made up of special type of alloy metal which doesn't react to the body. Okay, so likewise, that's what the research is done and the processes are made accordingly. Okay, so the inflammatory response is, they causes inflammation whenever there is any sort of prevalence of infection in the body. Okay, then antimicrobial proteins and the fever. Fever is a first sign of infection. So any sort of infection present in the body, the temperature in the body will increase. Okay, let's see the macrophages. They are the monocyte cells. Okay, they are present in the alveolar sacs as well as in the Kupffer cells. The Kupffer cells is present in the liver. Okay, it is found by a person called von Kupffer. Okay, so this Kupffer cell and the hepatocyte cells are present in the liver. They are acting as a macrophage, so they go and swallow. Likewise, alveolar macrophages, they swallow the bacteria that is present in the alveolar sacs. Okay, and also the microglia, it is a connective tissue cell that is present in the brain. They also help in the process of killing the bacteria. Okay, the neutrophils, they are the phagocytic cells. They go just simply swallow the bacteria and they also die. Okay, so this is a summary of this thing. So we discussed phagocytic action cells, the natural killer cells, inflammatory response, and the antimicrobial proteins, that is the interferons. Okay, alpha, beta, and gamma interferons are present. Okay, so let's see the phagocytic activity. You see, this is the bacteria. Okay, so what happens, these neutrophil cells, they go and swallow them and imbibe, that is pull the bacteria inside the cell, inside cytoplasm. Okay, now the lysozymes that is present in the cytoplasm goes and attacks this bacteria and hydrolyze them, kill them and neutralize them. So once it is neutralized, it is thrown out. So this is what we call it as phagocytic process. Okay, this is what we call it as phagocytic process. So that's why when there is prevalence of infection, more number of neutrophil cells keeps multiplying. Okay, body itself will produce more amount of WBC cells. So when you take the blood sample and see there is more number of WBC cells, it means there is some prevalence of infection in the body. Okay, coming to the natural killer cells, they are large granular lymphocyte cells. They they induce apoptosis. It is programmed cell death. Okay, so in cancer cells and viral infected cells also secrete potent chemicals that enhance the inflammatory response. Okay, so in this, when you are a child, your cells are good and it keeps multiplying. So as you grow, your epithelial cells keeps multiplying. For example, if you don't wash your body for 1 month, what happens? The epithelium becomes thick. So what happens when you take bath with water? You scrap and all the old cells keep moving. Likewise, this kind of cell multiplication process is taking place inside the body, but automatically they are programmed to be killed. That is what we call it as apoptosis. Okay, that's what we call it as apoptosis. Okay, next is the inflammatory response. So whenever there is an accident or trauma, okay, what happens? There is inflammatory process. Okay, so this inflammation is produced in order to prevent the invasion of bacteria, virus, and the fungus. I will tell you. See, for example, whenever there is a bone fracture, the muscles around the bone fracture site goes in for severe spasm. So this is also one of the defense mechanism. Okay, so whenever there is a bone fracture, there is inflammatory process that is taking place in the process of healing. So this inflammation will lead to formation of microscopic blood vessels and the formation of callus tissue. If the inflammation is not present in the body, then the healing doesn't take place. Okay, so there are some benefits. The beneficial effects are: prevents the spread of damaging agents, disposes the cell debris and pathogens, alerts the adaptive immune system, and set the stage for repair. So this is what the responsibility of inflammation is. If any wound, only when the inflammation starts, the neovascularization, the microscopic blood vessel will grow and then the wound will start to heal. If the inflammation is not starting, then the wound is not going to heal. Okay, the wound. This is a chart diagram how the innate defense mechanism is taking place. Okay, so whenever there is a tissue injury, what happens? There is heat, then heat followed by the hyperemia, that is the redness. Okay, then there will be severe amount of pain and swelling. This swelling is due to inflammation. So all this process should take place. Then only the healing process will continue and the tissue will repair. Otherwise it is not. So inflammatory response is very important in the body for any sort of healing process. Okay, it may be the bone or it may be in the skin wound, abrasions, anything. Okay, so now let us see the inflammatory response. It is one of the cardinal sign of inflammation is heat. You can see whenever there is injury, there will be production of heat, then there will be production of redness. Okay, then redness is followed by swelling. What happens in swelling? The intracellular fluid. See, two type of inflammation due to accumulation of extracellular fluid, we call this as edema, or inflammation due to accumulation of more intracellular fluid, we call it as swelling. Okay, so then there will be pain and loss of function. So these are all the cardinal signs of inflammation. Okay, redness, heat, swelling, pain, and loss of function. These are all the cardinal signs of inflammation. Okay, so coming to the inflammatory response, the macrophages and epithelial cells of boundary tissues, example GI and respiratory tract, bear toll-like receptors. They are the toll-like receptors. Okay, then there are 11 types of toll-like receptor that recognizes specific class of infecting microbes. So whenever there is injury, the receptors, they identify the bacteria. Okay, then these bacteria are transported through the lymphatic channels, and the lymphatic vessels will collect this in the lymph nodes. So, okay, this is at the tissue level, but at the cellular level, inside the cell cytoplasm, these bacteria are collected and stored in the vacuoles. Okay, So this is what happening in the inflammatory response. Then then the inflammatory chemicals, these are the inflammatory chemicals, that is histamine, that is released by the mast cells. Then proteins, that is circulating in the blood, the immunoglobulins, the IgA, IgM, all these are the proteins, okay, that is circulating in the blood. Then kinins and prostaglandins, that these are the chemicals that helps dilate local vessels, blood vessels, make capillaries leakier, then attract white blood cells. And the kinins and prostaglandins have sensitizing effects that causes the pain. Okay, generally with this prostaglandin we call it the endorphins. The endorphins will be released in the inflammatory site, and that creates the pain response. Imagine if there is no pain, what will happen? The patient will not know that the patient is having some wound. This is what happens in the diabetic peripheral neuropathy. So in diabetes, the peripheral nerves will stop functioning, and the response of taking the pain impulse to the brain stops. So what happens? These patients, they walk, have small injury in the leg, but they don't know. Then after few days, when you open, the whole leg becomes yellow in color. We call it a slough, the dead tissue. You keep removing, it goes deeper and deeper, but the patient doesn't feel any pain. Okay, so from this you have to know that pain is very important in the human body to heal the part. Likewise, inflammation process is very important. Okay, these are all the basic things the human being needs. Likewise, fever is very important to know that there is some sort of infection that is prevailing inside the body. If no fever, you will not know that there is no fever. Okay, so what happens? This causes vasodilation, causes hyperemia, that is redness and the heat. Then increase in capillary permeability leads to swelling and the pressure also. Then exudates, they contains the proteins, clotting factor, and antibodies. So here you need to know the difference between transudates and exudates. Okay, so once you open the abdomen and again do some operation and repair, here the fluid will leak into the peritoneum. Okay, so this fluid will be of two nature, exudates or transudates. So what happens if it is exudate, which contains more protein, clotting factor, antibodies, they stick the intestine each other and cause adhesions. So after the operation, the patient will be continuously suffering with stomach pain. So this formation of adhesion inside the peritoneum after the GI surgery is very big headache. Okay, so if it is a transudate, which doesn't have any, both are serous fluid, but the transudate doesn't have any protein substance. So what happens once this transudate leaks into the peritoneum, it doesn't stick the intestine each other. But if it is of exudate nature, then the intestine stick each other and cause severe discomfort for the patients. Okay, after the abdominal surgery. So that's why never try to open the abdomen unless there is a severe need. Okay, fine. So this is the difference between transudate and exudate. You will see that. Then let's see about the phagocytic mobilization. What is phagocyte? The swallowing. So once the bacteria swallowed by neutrophils, then as you have seen, the bacteria are neutralized by the lysozyme and it is digested. Okay, so this is what we call it as the adaptive immunity. The lymphocyte and the antibodies arrive at the injured site. Okay, now I have shown you the steps of phagocytic mobilization in the previous picture here. So this is the step of likewise, you see, neutrophil come, swallow the bacteria, then lysozyme attack, then they start to hydrolyze them. Then both bacteria and this cell will die and they will be released out. Okay, this is the phagocytic process. This is a mechanism of the phagocyte. So that's what leukocytosis means, increase in leukocyte, white blood cells. Okay, then marginalization means, so what happens, the neutrophils go and stick to the cells, bacterial cells, and then diapedes, that is neutrophil squeeze between the capillary cells, and chemostasis is taking place. That is previously what I have shown is taking in the cellular level, but this is phagocytic mobilization is taking place in the tissue level. Okay, so this is how the bacteria are getting killed. So in this war, what happens? Both the bacteria die as well as the WBC cell will die. So both these cells keeps phagocyting and keep dying. So this is what forms as a pus, that is abscess. Okay, so once the abscess is more, then you know that there is some abscess, you drain them out of the body. Okay, so if not, what happens? This abscess is large, so what happens? They communicate with blood vessels and they take all the infection all over the body, and they lead to septicemia. They lead to a condition called septicemia. Okay, so here you can see a foreign body goes inside, immediately the bacteria, then the inflammatory cells will go and accumulate here, and this is a foreign body. So the blood neutrophils, they detect that this is a foreign body, and they will come and swallow this bacteria, and bacteria die as well as neutrophil die. So likewise it keeps multiplying death, more number of cell death, and the abscess is formed. So at fourth day, when you press, this foreign body comes out as well as the abscess that is formed also comes out. Okay, so this is how you have to understand the process of healing. Okay, then the antimicrobial protein, that is the interferons and the complementary proteins, they enhance the innate defense body and attack microorganisms directly, hinder microorganism ability to reproduce. Okay, the interferons are the viral infected cells are activated to secrete interferons. Interferons enter neighboring cells. Neighboring cells produce antiviral proteins that blocks viral reproduction. Okay, see, whenever the tissue is open, anything can enter. Virus can enter, bacteria can enter, protozoa can enter, any organism can enter. So virus enters, the interferons goes and neutralize the virus. If bacteria enters, the neutrophil goes and kills the bacteria. Okay, if protozoa enters, then they go and keep multiplying. Okay, so this is what you have to under the interference. This is a mechanism that shows the interferon is going and neutralizing the virus. Okay, if virus is more powerful than interference, then the patient suffers. Then the patient will suffer. Okay, functions of interferon. So if you see, it's antiviral nature. It reduces inflammation, activates macrophages and mobilizes the NK cells. Then it is genetically engineered interferons for antiviral agents against hepatitis C and genital warts virus, multiple sclerosis, and hairy cell leukemia treatment. So for treatment for hepatitis C, then venereal warts, then multiple sclerosis, actually it is idiopathic. Virus infection is also a cause for multiple sclerosis. Okay, they use the genetically engineered interferons to kill these viruses. Okay, then major mechanism of destroying foreign substance, that is amplifies, then kills bacteria and enhances both innate and the adaptive defenses. So these are all the functions of the interferons in the body. Okay, so this picture you can see the complement activation picture, where the classic pathway and the active pathway works. So what happens, this is a cell membrane which is biphospholipid layer, and this is a gap junction. So what happens, these pathways, in this pathway, how this bacteria is imbibed into the cell, and the bacteria is killed. So this is what it is showing. Then let's go to the fever. Fever is one of the very important response in the body. Okay, it is showing that there is some microorganism invasion that is taking place. Okay, if the fever is not present, then you will not know that the microorganism is multiplying. Okay, leukocyte and macrophage exposed to foreign substance secretes the pyrogens. The pyrogen resets the body thermostat upward. Benefits of moderate fever, as I told you, by knowing the patient is having fever, there are a lot of benefits. Okay, causes the liver and spleen to sequester iron and zinc. Okay, then increase metabolic rate, which speeds up the tissue repair. These are all the two effects of fever. So not only this, if there is any local wound or injury, there is severe amount of high temperature that is present. Okay, so the advantage of the fever is the metabolic rate is increased, thereby more amount of antibodies are produced and more energies are produced. Okay, so this is a summary of the second line of defense, that is the innate cellular and chemical defense system. So I think we have seen this previously. Okay, now let us go to the third line of defense. Let us go to the third line of defense. The third line of defense, in this you have the adaptive immune system, then that protects against infectious agent and abnormal body cells, then amplifies the inflammatory response, activates complement. Okay, when fails, the AIDS and the cancer results. What is AIDS? AIDS is a acquired immunodeficiency syndrome, and what happens in this happen when the defense mechanism is unable to defend the virus. I mean, when the immunity is suppressed, then the HIV virus, that is human immunovirus that causes AIDS, will start to multiply. Okay, so there are some viral infections that can lead to cancers. Okay, for example, HPV. In the female cervix, there are HPV infections, human papilloma virus infection. This infection will lead to cancerous proliferation in the cervical os. Okay, that's why we generally, for female after menopause, we go for HPV screening. So once HPV, human papilloma virus, is positive, then the patient has to go for regular screening, because this viral infection can trigger the cancer cell proliferation. Okay, so in this you have the adaptive cellular humoral, and which is if the third line of defense is less, then this kind of disease will proliferate. Okay, now let's see the antigens. So what is an antigen? It is a substance that can mobilize the adaptive defenses and provoke an immune response. Okay, it is a substance that can mobilize the adaptive defense and provoke an immune response. So too much provocation of immune response is also bad. Okay, so for example, there are a lot of immune disease. For example, this rheumatoid arthritis, that's due to trigger of this antigens. Okay, immune response. Then we can give many type of this inflammatory joint diseases. Okay, likewise connective tissue diseases, all these are due to autoimmune response. Okay, so that's what you will understand here. So if you see, most are large complex molecules that normally found in the body. Antigens can be complete or incomplete. Okay, so what is complete antigens? It has two properties. One is immunogenicity and reactivity. Ability to stimulate proliferation of specific lymphocyte and release antibodies is called as immunogenicity. And reactivity, ability to react with products of activated lymphocytes and antibodies release. Example, foreign proteins, polysaccharides, lipids, nucleic acids, pollen grains, bacteria, fungi and virus. These are the examples of this complete. What do you mean by foreign proteins? For example, bee sting. For example, yesterday I had a sting in my hand. So what happens? That's a type of protein that is released by the bee. Okay, so it was sitting in the door. I just tried to open the door. It stung my hand. So now the sting will release some sort of protein substance, so that causes severe amount of reaction. So I have severe amount of swelling all over my hand now. Okay, so this is what we call it as foreign proteins. Okay, there the autoimmunogenicity and the reactivity is taking place. Okay, so likewise this takes place whenever there is any presence of bacteria, fungi or virus. Okay, now let's go to the incomplete antigens. We call it as haptens. They are small molecules. They are non-immunogenic by themselves or immunogenic when attacked to body proteins, causes the immune system to mount harmful attacks. Example: poison ivy, animal dander, detergents and the cosmetics. What is meant by poison ivy? It is a type of plant material. Okay, you can see it causes severe amount of immunologic reaction. Immunologic reaction. Likewise animal dander. Okay, likewise animal dander. So what happens here in animal dander? So for example, some people will have continuous asthmatic reaction. Some people will have continuous bronchial asthmatic reaction. What do you mean? What is that? So this kind of animal protein will go and causes allergic reaction in the body, will go and cause allergic reaction in the body. Okay, I mean in the respiratory tract. So what happens here? They trigger this autoimmune response and causes bronchospasm. Okay, likewise detergents. Some people, when they use certain amount of soap, they get more amount of itching. Okay, these are all the example of incomplete antigen attacks. Okay, so some people will be reactive to these kind of things. Okay, fine. Next, what is this antigenic determinants? It is defined as certain parts of an entire antigen that are immunogenic. Okay, example: antibodies and lymphocytes react, receptors bind to them. Most naturally occurring antigens have numerous antigenic determinants that mobilize several different lymphocyte population from different kind of antibody against it. Then large proteins, they are highly immunogenic and reactive. Then large chemically simple molecules have little or no immunogenicity. Okay, so here this is what you have. You can see here there are different types of models. Okay, to explain this antigen and antibody reaction, you can see these are the antigenic. These are different types of antigens. Okay, so so what happens? This antigenic determinants. This is has to come and lock with this. Okay, this cannot lock here. Okay, or this cannot lock here. So specific antigen has to determine specific antibody. Okay, so that is the model that is shown here. This is for your understanding. This is not the actual structure of the antigen and antibody, but know that specific for example, antigen A should come and attach only to this surface, not with this surface or this surface. Okay, this is what you have to understand about this antigen, that is identifying the antigenic determinants. Okay, next, the self-antigen, that is MHC proteins, the major histocompatibility complex. They are the protein molecules on the cell surface that is antigenic to others in transfusion or grafts. Example: MHC proteins coded by the genes of the major histocompatibility complex and are unique to an individual. MHC protein displays proteins, usually self-antigens. If infected cells, MHC proteins display fragments of foreign antigens which help mobilize the adaptive defenses. Okay, now let us go to the cells of adaptive immune system. The adaptive immune system cells are the B lymphocyte cells and the T lymphocyte cells, and also the antigen presenting cells. Okay, these antigen presenting cells are less in number. When you speak about the T lymphocyte cells, they are produced by the thymus gland, and B lymphocyte cells are produced by the bone marrow. Okay, the adult, 80% of lymphocyte cells are B lymphocyte cells and 20% of lymphocyte cells are T lymphocyte cells. Whereas in newborn baby, 80% of the lymphocyte cells are the T lymphocyte cells because they don't have bone marrow. only the B lymphocyte cells come from the mother will be circulating, okay, and 20% of the lymphocyte cells will be B lymphocyte cells in the newborn baby, okay. So here you can see the lymphocyte development, maturation, activation. As I told you, would have seen this in the hemopoiesis, that is B lymphocyte cells produced from the bone marrow and the T lymphocyte cells produced from the thymus gland. They go and migrate to the lymph node. In the lymph node you see the germinal centers, that is the place where they are maturing, okay, and proliferating. So these cells get activated once the bacteria enters into the body, and the lymphocyte cells will release these lymphocyte lymph node will release the lymphocyte cells, and they catch the bacteria and then trap them and again take it to the lymph node and neutralize the cells. That's why whenever you get any infection in the leg, your lymph node in the inguinal region will swell. If you get an infection in the throat, the lymph node in the neck region will swell, and after a few days it will reduce, because the bacteria are trapped and they are getting neutralized, okay. They originate in red bone marrow. The B cells and the T cells, when both cell types mature, they have immunocompetency and self-tolerance, okay. They are the naive unexposed cells are called B cells, and the T cells they are exported to lymph node, spleen and other lymphatic organs, okay. So this is what you have the description here in this picture. So if you see lymphocyte designed to become T cells that migrate to thymus and develop immunocompetency there. B cells develop immunocompetency in red bone marrow, okay, and they are maturing in the lymph nodes and then they are released into the tissues, okay. Coming to the T cells, the T lymphocyte cells mature in the thymus under negative and positive selection pressures. Positive selection pressures are nothing but selected T cells capable of binding to self-MHC proteins. Negative selection cells are prompts apoptosis to the T cells that binds to the self-antigens displayed by the self-MHCs, okay. These T cells, they ensure the self-tolerance in the body, okay. You know what? We are always surrounded by, we are always surrounded by the tuberculosis organism, Mycobacterium tuberculosis, okay. You know, this organism is present everywhere. Only because of the immunity we have, we are able to defend these cells and we are surviving without the tuberculosis invasion in the body. If our T lymphocyte cells suppression is present, then the tuberculosis organism will invade and start to destroy the organs, okay. So likewise, you know, for example, countries like India, they have more number of tuberculosis patients once upon a time, okay. So this tuberculosis is contagious and it can spread. Now it's almost controlled, okay. It's almost controlled, and there are good treatment for tuberculosis. You know, once the tuberculosis organism enters into the body, the patient has to take antibiotic therapy for around 9 months time, okay. This organism is very difficult to remove from, it's very difficult to remove tuberculosis organism from the body. So this is happening when there is T cells suppression, okay. So likewise, B cells, they mature in the bone marrow and they are also acting against the bacteria, okay. Now you have to know about this antigen presenting cells.